Okay, so now we're going to cover immunoglobulins, their structure and their function. Okay, so every cell, every B cell has a copy of the immunoglobulin genes, but it can express those genes in different ways. Okay, so on the one hand, it can express its immunoglobulin as a membrane-bound immunoglobulin, and that would generally be referred to as the B cell receptor. Okay, so the B cell receptor is just an immunoglobulin that is attached to the membrane. Of course, as well, the B cell can express immunoglobulin as a form of immunoglobulin that is secreted from the cell, and that's generally referred to as antibody or immunoglobulin. Okay, so I suppose the summary point there is that the B cell receptor is the exact same as immunoglobulin, with the only exception is that it's membrane bound. Okay, so we've covered that, and we're pretty much aware that antibody is produced by B lymphocytes. And we have to remember that any given B cell is destined to only produce one specific antibody or immunoglobulin against a particular pathogen, okay, giving it that specificity. Okay, so this is what an immunoglobulin will look like. Okay, you have two heavy chains and two light chains. Okay, the two heavy chains are the green section here and the other green section here. And of course, the light chains are the yellow sections. Okay, and the structures that hold these components together are interchain and intra-chain disulfide bonds. Okay, and you also have a hinge region as well, which we talk about. Okay, now in terms of the structural characterization of immunoglobulin, it's done in a number of ways, but we'll start with the Fab region. Okay, so the Fab region is the upper part of the immunoglobulin, and it contains the region that will directly bind to the antigen. Okay, whereas the FC region is the bottom component of the antibody, and this region is used to attach to phagocytes to enable it to eliminate any antigen that's bound to the immunoglobulin. We'll talk about this in a little bit more detail later. Okay, but at this stage you should appreciate the Fab region is the upper region that attaches to the pathogen, and that the FC region is for attaching to phagocytes. Okay. Now, in terms of the heavy chain, and in terms of identifying which components are, which components make up the heavy chain. So, on the one hand, you have immunoglobulins that can have three constant regions. Okay. Constant region 1 for the heavy chain, 2 and 3. And the important thing to also notice is the heavy chain has a variable region, which you can see highlighted here. Another thing that I want to highlight is some immunoglobulins, such as IgE and IgM, have 4 constant regions. Okay, 1, 2, 3, and 4. And the 4th constant region plays a part in the functionality of those specific immunoglobulins. Okay. Of course, the light chain has 2 constant regions. The light chain also has a constant region, and it has a variable region as well. Okay. And the heavy chain and the light chain are brought together and combined using disulfide bridges, and that is the structure of immunoglobulin. Okay. Now, there are 5 different types of immunoglobulin. You have IgM, which generally be associated with immunoglobulin response required for the blood. You have IgA, which is generally an immunoglobulin or antibody response that we need for mucosal surfaces. When I say mucosal surfaces, I'm talking about the lining of the respiratory tract or the lining of the gut. Okay. For IgG, generally speaking, they will be required for a general immune response, but they're particularly effective at reaching intracellular spaces. So given the small structure of IgG, it's very good for responses in hard to get to areas, if you like. IgD's an immunoglobulin subtype that you'll find associated with the B cell receptor, and it's a marker for immature B cells or maturing B cells. Of course, then we have IgE, which is particularly good at responding against parasitic infections and is also associated with some of the allergic reactions, allergies. So in terms of why we need different immunoglobulin types, we need to appreciate that, yes, you have different immunoglobulin types. Say, for instance, you need an immunoglobulin for Salmonella. The best structure of immunoglobulin that will be useful for useful against the Salmonella infection will be IgA. And the reason it will be IgA is because the structure of IgA is best suited to deal with mucosal immunity. Okay. And the only thing really that differs between when we decide we're going to have an immunoglobulin, a specific immunoglobulin type, the variable region won't change at all. So say, for instance, we have an immunoglobulin produced against Salmonella. The B cell then decides, okay, we have a B cell receptor with a variable region that will recognize Salmonella, so we won't change the variable region, but we need to change the constant region, because we need a constant region that's going to be best suited to reach the areas for a mucosal infection. And at that point, it will decide that it will have a constant region that is associated with an IgA. Okay. So when the B cells decide which immunoglobulin type they're going to select, it will be based on where the infection takes place, or certainly influenced by where the infection takes place. Okay. Now, immunoglobulin with 3 constant regions are IgA, IgG, and IgD. Okay, as you can see here. Whereas if we require an IgM and an IgE response, the IgM and IgE response will have a 4th constant region. Okay, and that'll be, for instance, if we need a rapid response in the blood, or if we have a parasitic infection. But IgM and IgE work in different ways, but the one commonality they have is they have 4 constant regions. Okay. Now, we talked about the differences between these types of immunoglobulin in terms of constant regions, but they can also differ based on how they're structured. Okay, so for instance, IgG will be produced as a monomer, that is a single immunoglobulin. As you can see, IgG has the ability to bind to 2 different antigens. IgE has 3 constant regions, as we described before. It's produced as a monomer, and it has the ability to bind to 2 antigens. IgD is part of the immature B cell receptor, and it has the ability to bind to 2 antigens. IgM is a different scenario altogether. It involves 5 monomers of IgM, and those monomers are joined together as one complex via a joining chain. And what this means is that IgM, when it's in this conformation, has the ability to bind to 10 antigens. So IgM is very, very effective at clearing up or aggregating a whole range of antigens very quickly. And IgA is generally released at the gut barrier, or the mucosal barrier of the respiratory tract as well, as a dimer, again with a joining chain combining them together. And again, the structure and the way these immunoglobulins are composed makes them suitable for different types of infection at different regions of the body. Okay, so when we're talking about differences between immunoglobulins, then we need to consider the number of constant regions and also the amount of monomers and how they're structured together. Okay, now in terms of the immune response, the type of infection will dictate whether it's IgG, E, D, M, or A. But one thing I should highlight at this point is that when we encounter an infection or an infectious agent for the first time, the initial response is always IgM. Okay, that's the default. Then the B cell, once it has more information on the pathogen, will decide whether it's going to change from IgM to one of the other Ig subtypes. Okay, and when it changes from IgM to either IgE or IgE, or it might decide to class switch to IgA, once it does that, that process is called class switching. Okay. Okay, and just to reiterate that this top region of the antibody is for antigen binding. Okay, these are the variable region, the sites that are specifically designed to recognize the pathogen that this immunoglobulin is targeted to recognize. The FC region, which is here, has a number of functions. One is to bind the FC receptors. Okay. Essentially what happens is the antibody binds to the pathogen, but of course it's opsonized the pathogen. That is, it has attached to the pathogen and made it easier for the immune system to recognize. But in order to eliminate the pathogen or the antigen that's attached to the antibody, it needs to interact with a phagocyte. How does the antibody interact with the phagocyte? It uses the FC portion of the antibody to attach to FC receptors on the phagocyte, and essentially what happens is the phagocyte will use its FC receptors to attach to the FC region, take it into its cells, break down the antibody, and also destroy the attached antigen. Another function of the FC region of the antibody is for complement activation. Okay, so for IgG and IgM, the FC region is a location where it can fix complement and activate complement. Another function of the FC portion of the antibody is for placental transfer. Okay, so when a woman is pregnant and they need immunity to be conferred to the fetus, the antibodies produced by the mother will arrive at the placenta, and its FC portion will bind to the FC receptor of the placenta, and that will enable it to move or transfer across the placenta. Okay, conferring short-term immunity to the fetus. Okay, so that's an overview of the structure of function relationships in immunoglobulin. We're now going to go on to focus on the functionality of I suppose, how antibodies are able to facilitate or eliminate a pathogen. Okay, so we've discussed IgM, ADG, and E, and this is just a summary of their uses. Okay, which you are free to read in your own time. Okay, and again, just to reiterate, the Fab region is the region of the antibody that's responsible for binding to the antigen. And the FC region contains highly conserved amino acid sequences. Okay, so this portion of the antibody is generally the same in a lot of immunoglobulins. And this region has no role in antigen binding. Okay, but it does anchor the antibody on the cell surfaces, particularly in relation to its binding of FC receptors on monocytes, macrophages, and other phagocytes. And another important role of this FC region is that It enables the antibody to bind, the antibody, once it's bound to the microbe, to act as a little binding site and for cells to attach, hence making it an opsin. What I mean by opsonin is it makes it easier for the pathogen to be identified by the immune cells. Okay, so here we have one functionality of the antibody is to directly bind and opsonize the pathogen, as seen here. Okay, so the Fab region, as you can see, has attached to the pathogen, where the FC region is like a little tail sticking out, trying to get the attention of the phagocytes. And below you can see where the antibody bound to the microbe then can attach to the FC receptor on the phagocyte as such. And in this case, the neutrophil has used this FC receptor to bind to the FC portion of the antibody, and that enables it to subsequently take the antibody-bound microbe into its cell, break it down, and essentially eliminate the pathogen. Okay, the next role for how antibody can contribute to immune response is through the complement activation process, classical complement pathway. Okay, generally what happens here is you have a soluble IgM complex which is not bound to the antibody, and in this conformation it won't activate complement, which is what we want. If there's no pathogen, we don't want that complement activation. But upon binding to the antigen, then that enables IgM to the FC portion of IgM to activate complement. Okay, similar with IgG, when there's no antigen bound, then it will not activate complement. Whereas once IgG has bound to the microbe, the FC receptor will enable activation of complement, and that's how antibody or immunoglobulin can contribute to the immune response in terms of complement activation. Another important function of antibody or immunoglobulin is for parasitic infections. Okay, so in this sense, what happens is if we have a parasitic infection, the B cell will class switch to IgE. The IgE will then bind to FC receptors on macrophages, mast cells, and eosinophils. And then once they're bound, if they then bind to a parasitic antigen, as seen here, as soon as the antibody binds to the parasite, it causes the release of lytic enzymes to destroy the parasite. Okay, generally speaking, what you would have covered in week 2 was that when phagocytes bind to a pathogen, they take them into the cell and degrade them through phagocytosis. Okay, it's just a standard process. The difference with a parasite is physically they're a lot bigger, and it's impossible to phagocytose them. For instance, a neutrophil could never phagocytose a parasite. It's just too big to take into its cell. So for a parasite, that's why we need IgE, and we need macrophages, eosinophils, and mast cells, because we need them to release lytic enzymes outside the cell to destroy the large structure that is the parasite. And as I say, one of the mechanisms or processes that enables this process to happen is through IgE mediated binding and release of lytic enzymes, antibody dependent cytotoxicity. Okay, and another way that IgE can mediate these effects on mast cells is that the IgE produced by the B cell upon first exposure, say for instance a peanut allergy, they will produce IgE. The IgE will then bind the FCE receptors on the mast cell, and they'll stay there for a duration of time. And then upon the second exposure, some of the peanut allergen arrives and binds to the IgE that's bound to the Fc receptors. And once the peanut allergen, just as an example, of course, there could be other antigens that cause a similar response. But in this case, the peanut allergy binds to the IgE bound to the mast cell. It cross-links, and once the antigen bound to the IgE cross-links together, it causes the mast cell to release its granules, and that causes mast degranulation, which causes the release of inflammatory mediators, which results in that allergic reaction, for instance, that you would get with peanut allergy. Now that's a situation where it's not a favorable outcome. But another situation where an IgE response like this is useful is for the likes of a parasitic infection. Okay, so that's how IgE immunoglobulin can contribute to the immune response. Another function of immunoglobulin is the neutralization. Okay, so pathogens or microbes have the ability to produce very toxic products. And one of the functions of immunoglobulin is one of the functions of the immune response is to produce immunoglobulins that will bind to these toxic compounds and neutralize them to prevent them having toxic effects on our bodies. And that's the effect of neutralizing antibodies, okay? So it's another mechanism where basically immunoglobulins or antibodies can offer protection against toxins by neutralizing them through binding. Okay, and this diagram here gives you a summary of the different functions of antibody and how they may contribute to the elimination of a pathogen. Okay, folks, and this is a lovely summary table about the different structure and functions of each of the immunoglobulins in terms of, what crosses the placenta, what fixes complement, what binds the Fc receptors, etc. Okay. Now, another thing that I wanted to cover was there are other components of immunoglobulins that help to impact their functionality. And one thing to consider are, if you can see, little carbohydrate moieties that can be present on the Fc region, which will influence their functionality. Of course, you also have the hinge region, which helps antibody have that sort of structure and degree of flexibility. And then also you have the variable regions as well. Now, another caveat to all of this is there are other subclasses as well. IgG has 4 subclasses: IgG 1, 2, 3, and 4. And again, it's based on the different versions of the heavy chain. And IgA also has subclasses: IgA alpha 1 and alpha 2. Now, in terms of the light chain that we discussed earlier, there are 2 types of light chain. There is the kappa light chain and there is the lambda light chain. So in terms of specific information on the antibody subclasses, so for IgG, it's a monomer, as I discussed earlier. And generally speaking, what you'll find with IgG is 66% is IgG1, 23% IgG2, 7% IgG3, and 4% IgG4. Okay, and this is just an example of the structural differences between them. You can see one of the main differences between IgG1 and IgG3 is the length of the the length of the hinge region. Okay. In terms of the properties, it's the major serum immunoglobulin, and as I said earlier, it's a major immunoglobulin in the extracellular spaces due to its small size. It's the sole contributor to placental transfer of antibodies to the fetus, and it does this through the Fc receptor. And IgG2 is generally poor at has poor uptake in the placenta. The subtypes of IgG have differing capabilities of fixing complement or initiating complement. For instance, IgG2 is fairly poor at fixing complement, and IgG4 cannot activate complement. In terms of attachment to Fc receptors, IgG2 and 4 are poor at attaching the Fc receptors. In terms of phagocyte synopsonization, and of course IgG is useful for antibody-dependent cell cytotoxicity in terms of binding the Fc receptors on natural killer cells. Now IgM, as we know, is a pentameric structure. So we have five antibody monomers attached together using a J chain. And of course, be mindful as well that it has 4 constant regions. It's the third most common serum immunoglobulin. It's the first immunoglobulin to be made by the fetus and B cells. And of course, it does fix complement. So this has been covered. And again, one of the main properties for immunoglobulin is the ability to agglutinate a large amount of antigen very quickly. And in terms of the first receptor to be expressed on a B cell, it's IgM, which you'll see here. The B cell receptor is generally IgM, and it contains CD79A and CD79B, which help to transduce intracellular signals once the antigen is bound to the receptor and cause activation. Okay, now IgA, as I say, is generally found on mucosal surfaces. Excuse me. And it's generally secreted as soluble IgA, which is a dimer, and is joined together as a J chain. And as I say, it's a secretory piece holding it together as well. It's the second highest serum immunoglobulin. The majority of this will be found in the mucosa, tear, saliva, gastric, and pulmonary secretions. Does not fix complement, or certainly very weak at fixing complement. Attaches the Fc receptors on some cells if they have that specific receptor. IgD is one of the ones that we probably know least about. Fourth highest serum immunoglobulin, and it does form part of the B cell surface immunoglobulin, and it doesn't bind to complement. Of course, Ig, which we've covered, has that 4th constant region produced as a monomer, and it binds to FCE regions on basophils and mast cells, and it doesn't require antigen binding. So these immunoglobulins will attach the Fc receptors without the need for antigen binding. And IgE is one of the main reasons why we get allergic reactions, such as food allergies, as I used the example I used earlier, the peanut allergy. But in terms of an effective and protective mechanism, it's useful for fighting parasitic infections through the help of eosinophils, mast cells, etc. And it does not fix complement. Okay, and this is the high IgE binds to mast cells upon release, as I covered earlier, and that subsequently, when antigen binds to these IgE on the mast cells, you get the cross-linking, and that causes mast cell degranulation and activation of complement.